This is related to the molecular clock and other methods that we discussed that are used to trace origins and migrations, mixing of populations. More genetics than I obviously know, but it's worth discussing. This is again from the Scientific American book on Becoming Human that we refer to from 2013 that you can find using Google. So founder mutations are a special class of genetic mutations that often cause human disease and that enables scientists to trace the migration and growth of specific human populations over thousands of years. So it's again like using mitochondrial DNA as and molecular clock to go back and do that, or use haplotypes to see how populations migrated and mixed around. So history in a sequence in terms of overview. Founder mutations are a special class of genetic mutations embedded in stretches of DNA that are identical in all people who have the mutations. So these are haplotypes that are getting passed around. Everyone with a founder mutation has a common ancestor, the founder, in whom the mutation first appears, which is obviously random, and it may be maintained because it's beneficial, as we will see in a minute, and eventually over time it may become deleterious or cause illness. By measuring the length of the stretch of the DNA that includes the founder mutation and by determining who currently carries the founder mutation, scientists can calculate the approximate date at which the mutation first appeared and its route of dispersion. Both pieces of data provide information about the migrations of specific groups of people through history, which is what we are discussing here in terms of migrations. As discrete populations mix, disease-causing mutations now associated with specific ethnic groups will be found more randomly. So in general, let's say Africans are more susceptible to sickle cell disease, which we'll discuss. Then they mix with other populations. Then the doctor has to look at the DNA instead of just relying on the ethnic group from which they come. Future medicine will turn to DNA analysis to determine risks of disease currently associated with ethnicity. Like Asians in the US, doctors note that they have a higher propensity for heart diseases, diabetes, and so on. So that's ethnicity-based precautionary diagnosis, but then individuals from that ethnicity may be different as well. So an old original versus numerous newcomers. This is how it works. You have a normal sequence and a mutation, and you can see that a mutation happened here that can be what is called a hotspot mutation chromosome, or you can have a founder mutation where in the normal sequence one of the T's got replaced with an A as a mutation, and then that gets carried forward. So you have to look at the structure around it to determine whether it's a hotspot mutation or a founder mutation. Okay, so if a group of patients with the same disease all had the same mutation at a given spot in their DNA, how could the physician know whether they were looking at a hotspot or a founder mutation? They could tell by analyzing the surrounding DNA sequences. Hotspot mutation could be like exposure to a chemical in the environment, or a pesticide, or some regions have high incidences of cancer that are not always understood well, and so on and so forth. Suppose that in all patients the code at one spot changed from a T to an A here. If A were a founder mutation, the surrounding sequence in all patients would be identical. The patients would have inherited the full sequence from the same distant ancestors, however far back it goes. But if A were a hotspot mutation, having occurred spontaneously at a place where the DNA is prone to error, the surrounding sequences would also show other differences, like we see here in gold colors. So the red versus gold, you can see that the structure around changes and not just that one letter that got mutated. The surrounding sequence would also show other differences at sites where DNA codes normally tend to vary without causing disease. Sickle cell disease, marked by misshapen red blood cells, this is what this is showing, is usually caused by a founder mutation. Achondroplasia, a form of human dwarfism here, ordinarily results from a hotspot mutation. Okay, so this evolved over time that protected the people with this one mutation to be immune to malaria, which is carried by mosquitoes, but then if you get two of those, then you get the sickle cell disease. Okay, getting shorter with age. A uniquely identifiable chromosome region, which we called before as the haplotype, that surrounds a founder mutation gets shorter over generations as chromosomes mix in a process called recombination. So when two people have sex and they produce an offspring, there is a recombination happening. In this example, the yellow chromosome in the founder holds the founder mutation, and the blue chromosome come from a normal parent. So you have one parent carrying the founder mutation marries another parent who is not carrying this mutation, and they produce a child that is carrying a mix now by recombination. When the founder produces sperm or eggs, the two chromosomes exchange sections. So as babies are born and they grow older, cells have to reproduce as we age, and that is mitosis, where the 23 pairs of chromosomes are replicated, whereas the one of the chromosomes out of 23 pairs, which are inherited from each parent, is what determines sex. So males have an X and Y chromosome in the sex chromosome, and females have 2X. And when females produce eggs and males produce sperms, there is a recombination. They have to split into having a pair of chromosomes into single chromosomes, because the gametes, sperm and egg, have to meet the partner. A sperm has to meet the egg, and they have to combine to produce again pairs of chromosomes. So the gametes, or the sperms and eggs, have to be so-called haploids. They cannot be diploids. So during that time, there is a combination of mitosis that happens in multiple steps. Okay, so that's where carrying offspring inherit a newly mixed chromosome that includes the mutation and other parts of the founder haplotype, orange region here. So chromosomal mixing over generations inevitably leads to a shortened haplotype. So let's say there is a founder who has had a mutation which is in one chromosome, and there is a normal chromosome because you have pairs. So mutation arises and recombination happens, and when the person marries another person that is not carrying the mutation, then recombination carries the mutation, but now the length of this chromosome that was carrying the mutation has been mixed to produce a shorter chromosome, and carrier grandchild will have even a smaller piece, and then the present day carrier over time will carry the mutation in one of the chromosomes as a shorter piece. Okay, it's kind of normal. Yesterday's genes become tomorrow's medicine. So in general, when there is ethnicity and there is known mutations, founder mutations carried like the sickle cell mutation, then usually that can be used to make precautionary assessment of the potential for a disease, for example. Observing ethnicity is currently a quick way for physicians to estimate the risk of certain disorders. As humanity's DNA becomes ever more mixed, the DNA itself will inform the doctor of an individual's predisposition for those diseases. So as ethnicities keep getting mixed with globalization and migration and so on, even not just in the Holocene but even before, then this recombination will begin to make shorter chromosomes of the founder mutation. So founder mutations may be viewed as special cases of a much larger group of disease-causing variants of our DNA. So what may have started as a DNA that may be beneficial, like a mutation in hemoglobin that gave you immunity to malaria, may end up making you sick when you get 2 copies, or something that like lower melanin in your skin when you are in high latitudes, and then you migrate to Australia, that may begin to give you skin cancer and so on. So there are many examples we will look at where such mutations are carried forward, and then even though they may have been beneficial at one point, which is why they survived and spread through the populations, can then end up causing diseases. Okay, so balancing selection keeps potentially deleterious genes circulating. In regions with malaria, which is spread by mosquitoes, having a single copy of a mutation in the hemoglobin gene is protective against malaria. Individuals with that mutation have higher survival rates against malaria, but those who inherit 2 copies of the mutation, let's say from each parent, suffer from sickle cell disease and have lower survival rates. So that hemoglobin mutation ends up being an issue. The competing forces lead to a stable level of sickle cell mutation in the population. So those who are carrying 2 copies end up dying, and that gene is not passed on. So you reach some kind of a balance over time to maintain what is a non-deleterious mutation. This is the biggest advantage of sexual reproduction. In bacteria and viruses that just multiply, they can multiply fast and grow in numbers, but a mutation can also quickly multiply and destroy a population, whereas in sexual reproduction this recombination can only begin to select beneficial mutations and get rid of deleterious mutations along the way. So noteworthy founder mutations here, I won't read the gene names, but conditions like iron overload, cystic fibrosis, sickle cell disease, blood clots, alcohol toxicity, some ethnicities cannot digest alcohol, lactose tolerance, I've probably heard of lactose intolerance a lot, deafness and dry earwax. The mutation origins are very different as well. Far northwestern Europe for iron overload, which may have started as a good thing, but then it became too much. Cystic fibrosis in Southeast Europe and Middle East, sickle cell disease in Africa, Middle East, blood clots, Western Europe, alcohol toxicity in Far East Asia, lactose tolerance in Asia, deafness in Middle East, and dry earwax in Northeast Asia. Migration routes associated with those: south and east across Europe, west and north across Europe, to the New World from Africa and Middle East. Worldwide now blood clots, north and west across Asia, west and north across Eurasia, west and north across Europe, outward including Amerindian populations for dry earwax. So possible advantage of having 1 copy of the founder mutation would be protection from anemia, protection from diarrhea, protection from malaria, protection from sepsis, infection, protection from alcoholism, possibly hepatitis B, allows consumption of milk from domesticated animals, deafness, this is unknown in terms of advantage and why it survived, and dry earwax, decreased perspiration, beneficial in cold, dry conditions. So this is a quick way of saying why this mutation would have survived over time. So in terms of uncommon origins of sickle cell disease, people have the same mutation from very different regions, but that mutation can occur within 5 distinct haplotypes, groups of genes from the chromosome that get inherited and create groups of haplotypes of populations, indicating that the mutation arose independently 5 different times in human history, as indicated in areas on the map. Patients can have the Senegal, Benin, Bantu, Arab India, or recently discovered Cameroon haplotype. 8% of African Americans carry at least 1 copy of the sickle cell mutation, which should help them against malaria, and survival rate should be high, which means they keep spreading it as opposed to those who have 2 copies and don't survive. So there is one population, Bana, Cameroon, Bantu, Arab India, and so on and so forth. So it gives you a sense of how complicated even the genetic information is in terms of migrations of our ancestors and mixing, and how those can be traced today in terms of figuring out common ancestors, migration paths, and mixing of genes, but also different illnesses or propensity for illnesses that different ethnic groups carry even now because of the founder mutations. Okay, so let's leave it there.